Good evening. Thank you, everyone, for sticking around. I'm from Breastlink. I'm a breast surgeon. Actually, we are a group of multidisciplinary specialists around Southern California, mainly in Orange County, and also moving up to the San Fernando Valley. So tonight it looks like California is well represented here. Our group has been interested in multi-gene panel testing for the past 2.5 years. The purpose of this study was really to look more at how cancers and the gene mutations we find are correlated, if at all. So this is a retrospective study looking at patients who underwent panel testing from July 2013 to September 2014. The majority of patients underwent a panel provided by Ambry with up to 43 cancer-related genes. All patients met NCCN criteria for testing, and all patients underwent pre- and post-test counseling. This study represents the first 500 patients in our practices who underwent multi-gene panel testing. We found the majority of patients did not have any mutations, where 6.4% of our cohort, or 32 patients, did carry a pathogenic variant. The pathogenic variants are shown here. Majority of them are BRCA1 or 2, followed by CHEK2, ATM, and PALB2. When we look at the patients with mutations and the type of breast cancers they were found, about 60% of patients had an invasive ductal carcinoma. The majority of them were luminal-type, ER positive, PR positive, and HER2 negative at 73%. 2 patients had a triple-negative breast cancer. One was associated with a BRCA2 mutation, the other with a PALB2 mutation. We also found lobular cancer and DCIS in our cohort. The bar graph here shows different cancers broken down on the bottom on the x-axis, ductal, lobular, and DCIS, as well as ovarian cancer and thyroid cancer. The single stripes represent the different individual genetic variants that were found. As you can see, this is a small study, so we can't really conclude with any kind of trends in each gene mutation. But what we can say is with larger cohorts, such as with the PROMPT registry, we'll be able to start making some more trends and conclusions. What we can find today, though, is that when we use a panel, we're going to find more mutations beyond just BRCA1 and 2 testing. Most of the patients we found in our study were found to have a mutation and a luminal type of invasive ductal carcinoma. We believe that if we understand cancer biology further, we'll better be able to figure out how we do screening, at what age MRIs need to be done. For example, prevention methods, for example, the benefit or indication for Tamoxifen or Raloxifene, and treatment algorithms. If we find more and more triple-negative BRCA-type cancers, maybe we can use platinums. We also believe here that surgeons and oncologists who are at the forefront of treatment should really be starting to talking to patients about the benefits as well as the limitations of panel testing. Thank you.